They fixed your house yet, Angela? We'll give it another couple of minutes. Ooh, get the fan going. See if anyone else rolls up. Oh no, that sucks. Well, tow truck is coming to pick up my car today, so it'll be a sad goodbye. Had a lot of fun in that car. Oh, I see. Well, that's a pain. As long as you get there eventually, right? Okay, who do we have today? Wow. That's a long time. I bet you know everybody at the desk really well by now. Hopefully you've got a like a room with or a suite, right, with a kitchenette or something like that. It's not just a room with a bathroom or something, right? Well, that's something at least. Imagine having takeaway every day can get kind of old after a couple of months. Oh, let's get that a little bit closer. It's probably just like being I remember when I was a grad student, we lived in a 2-bedroom apartment for a year or two, my wife and I. And then we had a studio apartment. Don't know if I've told you this, but it was so small that we had a hamster called Captain Nemo for some reason. Can't remember why. And we had to put his cage in the bath because there wasn't any room in our bedroom, living room, dining room to put it. So every time we wanted to shower, we'd have to take Captain Nemo in his cage out of the bath. Yeah, I know. And stick him in the living room. We had a futon, which converted from our couch into our bed every day. It was interesting. Okay, all right. Where are we at today? We are finishing up control of gene expression. So let me bring that up for everyone and me. So we're going to bounce around in this PowerPoint a little bit, and there's the chat. So it's going to be a little bit Well, hopefully not nauseating, right? But if you feel lost or confused or whatever, just give me a shout and I'll retrace my steps so that you can kind of figure out where you are. So I'm then lost and confused after I started, not before. I can't do much about that. Actually, that's a good point. Given the last exams coming up next Tuesday, which is a little bit nuts as far as I'm concerned. Anybody got any questions about any of the stuff that we've been covering? And that can be bacterial gene reg, as well as what we're finishing off today. And really, like, there's plenty of time today. The second PowerPoint, which is Introduction to Biotechnology, isn't. It's probably going to be more of a ramble through down memory lane for me, kind of stuff. So we have time. So if you have any kind of stuff, like cast your mind back. I know it's a little bit intense, but if there's anything that you want to go over, then just ask. Histone modifications. Did you see the videos that I sent, the WeHi ones? Yeah, they're pretty neat. They don't have a lot of dialogue, right? They're pretty sparse in that front. They tend to use the imagery more than anything. Okay, so essentially I'm gonna quit the sharing and I'm gonna bring this up so it's a little bit bigger. Oh God, my back is bloody killing me. Unsurprisingly, obviously. Right, so we can have a look at those videos in a second. Let's do this first, just to see if I can add some context or some clarity. So essentially, you have two states of chromatin, and that is heterochromatin. I don't actually even know why it's called heterochromatin. I know it's called chromatin, but I don't know where the hetero bit comes from. And euchromatin. And I'm just kind of recapping this stuff. I know you probably already know this, but it's a really handy way of putting this stuff into context, right? And so, well, let's use different colors. That's always fun. So this is closed, and essentially no gene expression. EXPN is expression, just being parsimonious with space. Right, so this is shut down. Good example is the X chromosome. Oops, chromosome. X chrom in ladies. Run out of space. Got a tiny little bit of space to work with here. So one of those videos that I sent is all about X inactivation, which happens in every one of, if you're a lady, or even if you're just a woman, I don't know. Sorry, that was a bad joke. Every single one of your cells that is diploid, so your somatic cells, every single one has one of your X chromosomes, 1 out of 2, inactivated, right? So it's all heterochromatin. And then over here we have euchromatin, and I always used to make this joke. It's kind of getting worn thin a little bit now. The EU was really open to migrants. This was a few years ago. So euchromatin. Obviously, that doesn't work anymore because they're totally not open. But that's how I remember it. And so this, don't forget, this gene expression, this is conditional on other stuff, right? It's not that as soon as you open up chromatin, that inherently means genes within that stretch of DNA are expressed. It simply means that they are then available to be expressed because that DNA is now accessible to various proteins and so on and so forth. Right, so really it's all about kind of opening the door, you know, to a visitor and then slam it in their face. It's kind of like euchromatin versus heterochromatin. So let's think what would be the best way of doing this. Let's get some. Can't remember what colors they used in the videos, unfortunately, but let's see if this will work. Right, so basically you have, just kind of thinking about colors right now. I'm going to just draw histones as just one single circle, just to make life a little simpler. And I'm going to draw some of their tails. So one of those is a, oh man, my handwriting sucks with this thing. Hold this a little bit steadier. Right, so remember one of those is a nucleosome. And DNA is wrapped around those histones, right? Those histones proteins. So those little wibbly bits sticking off is your histone tail, right? Super, super important part for control of chromatin structure. Damn it, shouldn't put that back on. And it's really where all the exciting stuff, as regards to histones at least, happens. And so, let's use green. On those histone tails, you will find methyl groups or methyl groups, however you pronounce it, right? Which essentially they do two different things. One is they allow these nucleosomes to be packed closely together, right? So they facilitate this kind of like really dense, kind of tightly wound structure. That's one thing. The second thing is that they displace the acetyl groups or acetyl groups, which neutralize the charge on those histone tails, right? So it's kind of like in a long way around of saying it. When you have methyl groups attached to those tails are charged and that DNA is tightly bound, right? So that's kind of like the two, I'll hold my fingers that way, the two different things. One, DNA is tightly bound to those histones. Two, those histones or nucleosomes really, because it's protein and DNA, are very tightly organized, right? And so the overall consequence of that is that the DNA wrapped around these histones is not accessible, right? It's physically hard for other proteins to come in and attach or recognize and attach to that DNA and begin the process of transcription, right? Whatever those proteins are, transcription factors, RNA polymerase, stuff like that. Does that make sense so far, what I've put up? Cool beans. And so, and don't forget, one of the really important parts of all of this is that this is a dynamic process, right? It's not fixed. I mean, it is for the X chromosome inactivation, because you don't want that to kind of stop happening. But in general terms, this is not a static situation. And again, this is a fairly stereotyped way of drawing it. It obviously is, as with all things science, right, way more complicated than this. Right. Actually, you can kind of imagine that this DNA is a little bit more loosely attached. And so now, ooh, let's use red. Red. And again, bit of a simplification because these residues, these are typically lysine residues, amino acids. You can have multiple ones per tail, and you get all kinds of different patterns based on which ones are acetylated. Right. And so SADOL groups, basically, we want to kind of stress the point even further. Oops, these are supposed to be pluses. Right. In the absence of acetyl groups, these tails are positively charged, right? Which is why DNA sticks to them so well. When you add acetyl groups, you basically remove that charge, right? And so in doing so, that DNA is less tightly bound to the histones, right? So it's kind of like loosened up a little bit. And also, these nucleosomes can spread further apart. They're a lot more mobile, right? They can slide, they can rotate, they can even be removed, right, by those chromatin remodeling complexes. And basically you have a bunch of proteins which facilitates the dynamic switching between the two. And you can have intermediates as well, right? It's not an all or nothing, depending on how many of these tails are acetylated versus methylated, right? And you have proteins called HDACs, which are histone deacetylases. Oops, gotta spell this right. And DNMTs, which are, I'm just going to call those methyltransferases. Can't off the top of my head remember what the DN part stands for. It'll come to me at some point, I'm sure. Right? Yeah, I will do it. And it also will come up clearer in the picture as well. I'm just trying to do it small because I run out of space. And then at the top, you also have hats. I can't remember. There's demethylases. I can't remember what the shorthand for those is, which are histone acetyl transferases. Yeah, none of these words are short. Let me just type those out for you while I remember. So HDAC equals histone deacetylases. Oops. DNMTs. Ah, crap six. I'm going to have to look up what DNMT stands for now. Oh, that doesn't help people. DNA methyltransferases. Oh, that's easy. Actually, that might not be the right one. We're just going to rub D and N off of that because I think that's more about methylating DNA, which we're not really looking at here. Oops. And all of those two together equals heterochromatin. Whereas hats are acetyl. And what's the, let's see. No, I don't have a, oh, HDM, there you go. Crab sticks, these things are not easy to type. There you go. So essentially, like, don't want you to stress too much about the actual words, but what I do want you to get, and I'll run out of space to put demethylazers up at the top, is this, maybe worry about HDACs and hats. There you go. Histone acetyl transferases, they're acetylate histones, they make it open and accessible, euchromatin, so that genes can be expressed. HDACs or histone deacetylases remove those acetyl groups and transform it into heterochromatin, right? And so really it's a continual back and forth between those two different states. Remember, methyl groups, whether of histones or of DNA, typically result in no gene expression, right? So DNA methyltransferases are involved in shutting down gene expression, typically via the promoters. Whereas acetyl groups on histones are associated with gene expression, right? Because they allow that more open configuration, which is more accessible to proteins, right? So back and forth between these two, and that's mediated by whether or not both actually the DNA and protein are methylated, the histones, and whether or not those histones are acetylated, right? Back and forth between the two. How about that? It's a lot of information and stuff, but really I want you to get like the general principle behind it. These details help you understand that, but really you need to understand this kind of dynamic switching back and forth between these two states, between heterochromatin and euchromatin. And that's managed by essentially what you do to those histones and the DNA as well. Did that help? If not, just ask questions. Okay, let me take a picture of that. Shit bits. Sorry, you're gonna have to just hang on for a second. I left my camera downstairs. Let me just take a picture. And the crazy thing is as well, I'll change my view in a little bit, is that DNA methylation also interacts with chromatin modifications, right? So there's a feedback loop between the two. So let's see. Essentially in DNA methylation, right, you can have I'm just going to draw these up like so, right? So that's the kind of standard notation for start of transcription, right? So there's going to be a gene down there, whatever that may be. Let's say, complete brain fart. I don't know, whatever that is, gene X. Just put up a question mark. I was trying to think of a tumor suppressor gene, but my mind's gone blank. So essentially in a lot of promoters, you have what are called CPG islands. Right, and those are really regions which can be methylated. Doesn't mean that they are. It just means that they are sites where methylation can occur. Right, now what color did I use for methyl groups? I think it was green. Okay. So if you have, make sure you're seeing this properly. Basically, DNA. DNA methyltransferases. What they do is they add methyl groups onto, onto the DNA. Unmuted? Don't believe so. Can you hear me now? Oh, please mute. Oh, I'm sorry. I read PI for some rather than please. Not very good at text speak, unfortunately, even though I've got a shitty old phone. And so if you add methyl groups to these cytosines in the DNA, essentially what you get two consequences, right? Transcription factors and maybe RNA polymerase. I'm not so sure about that. Well, that's DNA. They can't bind because these methyl groups physically change the appearance of this DNA. So where normally a protein would be able to come and recognize that region and facilitate turning on that gene, now because of those methyl groups, they cannot. And that leads to an increase in, my writing sucks, sorry peeps, histone methylation. Right, so those two things together equals no gene expression. Right. Now, obviously, you can reverse that with DMs or demethylases. I can write those out too. And that equals, or can equal, gene expression. Right. So that's another level at which you can control gene expression on a variety of scales, right? So chromatin modifications typically are a larger scale control of gene expression because they open or shut large regions of DNA for expression. DNA methylation is a little bit more specific. It tends to be gene specific, right? So it's really about the regulatory regions in that promoter. But it can also act as a feedback loop by affecting the methylation state of the histones that are interacting with that region of DNA, right? So essentially, once the DNA becomes methylated, then the histones will become methylated and deacetylated, and that will further shut down gene expression kind of in that particular region, right? Whereas the kind of the available state would be no methyl or methyl groups in this promoter. Obviously, it depends on whether or not that chromatin is open or not, whether that gene can be expressed. This doesn't necessarily mean that it is still able to be expressed, but now it's kind of available for expression if those other criteria are met. How about that? Makes sense? I'm going to type out those. Let me do some stretching. I'll type out those words just in a second. Which is DNMT. Well, I have a bunch of muscle relaxants and stuff from the ER at the weekend, so I might take some of those later. My wife says that they make her really groggy, so I thought that was the last thing that I'd want to take before teaching, because I'd be like, yeah, you know, don't think you really want to watch like 2 hours of me sleeping on my couch. I don't think that would be super exciting. Dang it. Yeah, I'm on Tylenol right now for my headaches. Whereas DMs is I think they're probably called DNA demethylases, so DDMs or something like that. But that's not super important. Yeah. Well, I can't go out and get it now, can I? I just have to suffer through for a little bit. There you go. So again, as with all of this stuff, it's a complex interplay of lots of different things that are happening, right? So one thing alone does not equal lack of gene expression, and one thing alone does not equal the opposite either, right? So one big kind of picture thing that I want you to get is that there are an awful lot of different things interacting to create one output, right? Either lots of gene product, functional protein, or no functional protein, or functional protein here, but not here, or this time, but not this time. All of that jazz, right? And so it's not just one particular thing. There are lots of different layers at which information comes together to control that, right? And that could be signals which are extrinsic to that cell, right? They come from outside, like I don't know, whatever you might think, like got headaches, right? So it's basically some part of me telling some other part of my brain that, hey, this isn't good, right? You want to do something about that. It could be signals from within the cell itself involved in, maintaining certain status or changing status, right, depending on exterior conditions, right? So there's lots. It's a very, very fine-grained decision-making process, essentially. It's kind of like comparing, I don't know, some of you are probably too young to remember these, but like old Intel 386s, right? You know, that had like 50 transistors on a chip or something like that, to modern processors, right? Which are, you know, have billions and billions of logic gates on their processors, right? You know, you could think of prokaryotes, not meant to be like derogatory about bacteria or anything, but they have a much simpler decision-making tree, right? Do we have anything? Do we need it? It's kind of prokaryotes. Eukaryotes are, where am I? What's the context? What time is it? What's the external conditions? What's the internal conditions? What other things are going on? What things are needed like in the future, right? There's just a whole lot more information that comes in, and that creates ever greater subtleties in how these things work. It may be that it's not subtle at all, you know, you just get boom, gangbusters amount of protein, but that's not always the case. It's usually extraordinarily complex. Okay, any questions about chromatin and DNA modifications before we move on? Cool, cool beans. That was fun. So let's pop up here. I wonder if I can make that smaller. I can. Okay, so I'm going to be bouncing around in here for a bit because, again, it's originally from a different professor, and I don't know how he teaches it, but all the information's here. It's just not necessarily in the order which I would present it in. So really, once we've got through, if we go back up here, come on, right, we've really spent a lot of time talking about this, right, because it's a really important aspect of gene expression in eukaryotes, right? What I want to do next is talk about control of transcription, right? So essentially, we go here, move this over a little bit so I can see what I'm looking at. And every eukaryotic gene has this kind of stereotyped regulatory region. There's always a core promoter, right? This is where RNA polymerase, or it's called the basal transcription apparatus, all the other bits and pieces, right, which are necessary to get RNA polymerase active and transcribing that gene. Those bind to that core promoter, quite often via the TADA box, right, the TADA box binding protein. And there's an awful lot of other stuff going on there. It's a really big, complex protein. There's also a regulatory region, right? And this is really where a lot of the complexity in eukaryotic gene regulation exists, right? So this core promoter, they're typically, I'm trying to think from memory, maybe 300 to 500 bases long, right, from the transcription start. So they're pretty small. And typically you can express a gene, maybe you want to use a green fluorescent protein as a reporter, using just that core promoter, right? It's not necessarily going to express very well, but it will express, right? It will work. However, to get finer control, so some of that control is mediated by these transcription factors, right, which bind specific sequence in this core promoter. But if you want to have much finer control, particularly over amount as well, then this regulatory promoter comes into play. And so this regulatory promoter can vary hugely in size. Doesn't even have to necessarily be next to the gene itself either. right? So you can have, it could be a few hundred bases, could be a few thousand base pairs in terms of DNA sequence. It could be millions of base pairs away at the other end of the chromosome, right? It's a much more flexible concept than this core promoter. This core promoter is always right next to the, or upstream of the transcription start site. In eukaryotes, at least, it always has this title box, right? There will be sequences that transcription factors recognize. The regulatory promoter is much more of a nebulous concept. It's actually astonishingly hard to figure out the true extent of a gene's regulatory promoter, right? Because they can be just huge, right? And there's limits to how big of a chunk of DNA you can clone, essentially, in front of a reporter. Okay, and so these regulatory promoters are typically where transcriptional activator proteins, enhancers, silencers, and other proteins which modulate, right, that gene expression, right, either to dampen it down or to, you know, amplify it, depending on conditions, right? So keep that in mind. Core promoter is where the RNA polymerase holoenzyme and all the other jazz assembles, and then your regulatory promoter, which is a much larger, more nebulous region, which controls quite typically, actually, the magnitude of expression. Okay, scroll down here. Nice big jump. Cha-cha. Okay, so essentially what, let's make that a little bit smaller. What we're talking about are things called cis regulatory elements, or CREs, right? These cis regulatory elements are, let's go way back up here, right? Elements that are potentially within the core promoter, but typically within this regulatory promoter, right? These are basically DNA sequences that are recognized by proteins. That's actually almost always the case in terms of transcriptional regulation in eukaryotes. It's always about what DNA sequence do you have here, what things can bind to it, and what things are present that can bind to it, right? That's the three different things, maybe two. Okay, so just kind of giving you some language, essentially, right? So enhancers are, as the name would suggest, typically about enhancing gene expression, right? Remember, boosting expression of stuff, you know, basically activating things that are bound to the core promoter. One way of thinking about this is like the catabolite activator protein, right, in the lac operon. Right, it doesn't necessarily recruit RNA polymerase to the core promoter. It just means that it's more efficiently bound and activated, right? That's really what enhancers do. We have silencers, right? And those are, as you would guess, involved in silencing gene expression. They often do so by, let's go back up here again, binding to and occupying those enhancer sites quite often. So they prevent the activation of transcription by preventing the binding of these transcriptional activator proteins. What you'll tend to find is that these DNA sequences are kind of scattered around in there, and they quite often overlap. Not necessarily the sequence themselves, but the proteins that bind to them. If you have one bound, you can't have another one bound, right? So that's another way in which we can play around, essentially. We have insulators, right? Insulators are essentially ways of separating one gene from another, right? Because these bits of DNA which are involved in the regulation of different genes can be far away. You don't want an enhancer bound to, or activated protein bound to one enhancer to activate transcription from a different gene, right? You need these things to be fairly specific, right? So if there's a likelihood of that happening, then you'll typically find what's called an insulator between those two genes, so that when an activator binds to one regulatory promoter, it doesn't accidentally activate or enhance expression from a different gene. And these response elements are really just a fancy name for saying DNA sequences that are recognized by proteins, right? And so they have a specific sequence, and that shape and sequence and that kind of topography are recognized by proteins, right? And really, in many ways, it's the accumulation or change in these response elements that has driven the change in morphology and complexity that you see in through the course of evolution, right? So one really good example, maybe we'll get time to talk about it, maybe we won't, is the evolution of wing spots in butterflies, right? And so you have butterflies that don't have wing spots, or that they have wing spots without, like the shiny eye, like the whole manga white dot in the eyeball thing. Essentially, the butterflies that do have them have accumulated a response element upstream of a gene which controls that feature, right? And so these are little DNA sequences which don't obviously encode protein, but they can have a massive difference in terms of how that gene or how much of that protein is produced and where. Anybody got any questions? All right. So here's just a simple graphic, basically, right? So all of these genes have core promoters, right? And essentially, we have three different genes here. SV40 is a virus, DNA virus, and it has astonishingly high levels of gene expression, which kind of makes sense because as a virus, you want to be producing viral proteins and whatever's necessary to form virions as quickly as possible, right, before the immune response has a chance to come and shut down the party. And so typically, actually, if you want to produce a lot of protein in cell culture, say like yeast or fly cells, something like that, most of those protein producing constructs will use, unless that protein is toxic, will use one of these SV40 promoters, right? So as you can see, there's lots and lots of these GC box sequences, right? So presumably there are activator proteins which recognize and bind that sequence, and in do so activate expression at a high level. Now, when you start looking at other kinds of proteins, right, we've got two very different genes here. We've got kinase and we've got structural protein, right, histone protein. And what you'll see is that they tend to share some of those response elements, right? We've got I don't know what these do. I really don't. Sorry. I know a tiny fraction of all that is to be known out there in terms of gene regulation. So I don't actually know what these do. But you can see that these some of these boxes are shared. You get different quantities, right? You may have other boxes present, right? So depending on which combination you have will dictate a whole bunch of different things. It will dictate how much expression you get from that gene, right? So one could predict, based on what I know about this particular promoter from a virus, that this thymidine kinase will be expressed potentially at a higher level than this histone protein because of the presence of those two GC boxes, which are responsible for the high level of gene expression from this virus promoter. Now, these other ones may be also involved in level of expression, but they also may be involved in location of expression, right? Or expression in response to certain conditions, right? So typically for any given gene, you'll have a bunch of different scenarios in which that gene can be expressed, right? And to different levels and in different places, right? And those different scenarios are controlled by these different boxes, right? Does that make sense? There's quite a lot to get in one go. But really it's like I don't know, they're like, I'm trying to think of an analogy. I was going to use cassette tapes, but most people don't know what those are anymore. It's really like small, interchangeable little bits of information. If you have these, then this is a potential consequence. If you don't have those, then that's not going to happen, right? And so these little bits of DNA are kind of, when you take them all together, you get the big picture of what that gene, where, when, and how much that gene is expressed, right? So it's all of those things together combined to give a particular output in a particular set of circumstances, I guess. One thing that is worth pointing out, so if you remember a little while ago, I talked about how in prokaryotes, they will group all of the genes together from metabolic pathway in one operon, right? So the trip operon is a great example of that. So that when you need to metabolize, produce tryptophan, I was thinking about lac operon for a second. When you need to produce tryptophan, you just express all of them together, right? It's like a package deal. We don't tend to have that in eukaryotes. Instead, what we have are these response elements. So say, for example, one of my favorite examples is the heat shock response, right? If you are, or your cells are suffering from elevated temperature, right? Maybe you're on the verge of heat stroke, all right? So your core body temperature is rising, bad stuff starts happening, right? And there's a lot of different things that you need to do to prevent that bad stuff from happening. One will be making sure proteins don't denature. Another might be making sure the cell membranes don't fall to pieces, right? Or become too fluid, right? There's lots and lots of different problems that associate with elevated temperature. And so there are lots and lots of different proteins that can help with that, right? I mean, it makes sense. And if you are suffering that, or if your cells, right? Because we're not doing this like directed or anything. If your body is suffering from heat stroke, then you want to have a coordinated response. You don't want to do like a little bit of this in one cell or one tissue, a little bit of this in the other one. You want to do it everywhere, and you want to do everything that is necessary. So all of the genes which are involved in a heat shock response have a heat shock response element, or HRE, in their promoters. So when temperature increases, basically the transcription factor changes shape, right? So kind of permissive temperatures, normal temperatures, it has a particular shape, doesn't recognize DNA, right? Doesn't bind to those heat shock response elements. As temperature increases, right, it is a heat labile protein, right? It's sensitive to temperature, right? Its shape changes, right? Kind of goes like that. And now it has the shape where it can recognize those heat shock response elements. And so now it's able to go and bind to those, activate transcription, and now you have your coordinated response, right? So again, in prokaryotes, they coordinate everything by sticking everything together. In eukaryotes, we have a coordinated response via these cis regulatory elements, right? These heat shock response elements. Now, those genes may be involved in other processes as well, right? Maybe like protein folding, you're going to have issues where you produce too much protein, right, at any one time. Maybe it's something that you need all the time, just at a low level, right? So the promoters for those genes are going to have other response elements in them, right? Some which may be present all the time, right, to activate transcription at a certain level. Others may be present under other circumstances, right, whatever those might be, you know, protein toxicity, for example, right? So that's how in eukaryotes you get that kind of coordinated response to a particular situation is through these cis regulatory elements. Okay, I'm going to do a little bit more and then I'm going to take a break because I'm pretty wiped before we start the next chunk. So another way in which you can do this is rather than just having different elements in that promoter, you can have 2 promoters or even 3, right, for any given gene. And I'll actually show you an example of this in a moment from work that I've done in the past. Right, and these are called alternate or alternative promoters. And basically they vary in lots of different ways. They can vary in where what cells have proteins that recognize them, right? So you might have a promoter for a particular group of cells and a different promoter for another group of cells, right? You may have an example we're going to look at in a second, a promoter that's specific to different life cycle stage, right? So larval versus adult or juvenile versus teenager, for example. And you can also have different strengths. You can have different parts of the gene in front of them, right? So if you express it from one promoter, you get like a little bit. And if you express it from another, you get a bigger bit. And that extra bit may have a different function potentially. And again, I'll show you in a second a good example of that. So this is a just kind of fairly standard kind of way of looking at this, right? So essentially you have a gene, what is that for? Does it give any? Oh, alcohol dehydrogenase, right? So a gene involved in turning ethanol into something less toxic, right? And so basically fruit flies have different life cycle stages, and those different life cycle stages eat different things. But one of the things that they do like eating is rotting fruit. And one of the things you often get in rotten fruit is alcohol, right? I mean, that's fermentation. That's how we get booze. And so they need some way of detoxifying that ethanol so that they don't get all tipsy and then can't fly anywhere. Or if they do fly somewhere, they're not like flying into windows and things. And so they have, Drosophila has 2 different promoters for that same gene. So you end up with 2 different pre-mRNAs, but you end up, in this case at least, with the same mRNA, right? So this doesn't change what form of protein you're going to get. It merely controls at what stage and in response to what you express that gene, right? That's so essentially it's kind of what we're talking about with cis-regulatory elements, but rather than having different elements within those promoters, you just have different promoters. It's kind of like a more absolute way of doing things. Oops, I'll do that. So let me see if I can share this. Ooh. So this is a promoter that I wrote. Here I am. When I was a postdoc at Penn State, this was a big chunk of work for me. And I'm going to use this example when we talk about biotechnology later, because there's biotechnology up the wazoo in here, and there's some cool stuff, pretty pictures. At least I think they're pretty. But the thing that I really want to illustrate here, and this will apply in a little bit, right? So this is a gene called PNC1. It's shit, what does it stand for? Pyrimidazolazine, blah 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 blah. I can't remember. It's a really long word. That's why I call it PNC1. But this is involved in essentially recycling a particular compound back into NAD, right? So you're familiar with NAD in terms of cellular respiration, right? It's one of the electron carriers that drops off electrons in electron transport chain. And so this is a protein that's involved in making sure that whatever that gets turned into, it gets back. And so this is some work that some work that other people did and some work that I did, right? So this, just to give you, because this looks kind of crazy, right? This particular gene has 5 exons, right? But only ever produces a mRNA transcript with 4, right? Because we have alternate splicing, which I'll talk about in a second, right? And here's the mutation, which I think I talked about a while back. It results in a small truncation and completely gets rid of enzymatic activity, so it's a null mutant. And this gene has 3 different promoters, right? It has a promoter up here in front of this exon. And this exon has what's called a signal sequence, which results in that protein, when it's made, being exported or secreted out of the cell, right? So it can float around and do something somewhere else, right? This one, 1B, kind of skips that, right? So it does, the protein produced from this promoter does not have that signal sequence and cannot be secreted, right? So anytime you get expression from this promoter, it results in a protein that can only work inside that particular cell that it's expressed in. If you get it expressed from the other one, then it can flow around inside the worm and do its metabolic business all over the place, right? So obviously now you have 2 very different scenarios, right? We have a case of, well, maybe we need to do a lot of this in metabolically active cells. We need to turn the gene on from this promoter, okay? But there are some cells where this gene is not expressed, right? So we need to provide this function to them externally, right? So then you're going to express it from this particular promoter. This one is a bit of a mystery, to be honest. Results in a different pattern of expression. Why it's there, who only knows, right? So this is a really good example of those multiple promoters, and also why you would have multiple promoters, right? Because this essentially allows a different protein to be produced from the same bit of DNA, right? So the DNA is not changing, the information is still the same, but now we're choosing which bits of it we turn into something functional. I think that's a pretty neat picture. We'll talk about some of this stuff down here later on after the break. Does anybody have any questions? No. Okay, so the final thing that I want to talk about, oh, actually I didn't mean to do that, never mind, is actually really neatly illustrated by that particular example too, is differential, yeah, we can talk about, very quickly, let's talk about alternative splicing too. Oh, we're actually going to, I better keep talking, we've got a bunch of stuff to cover, kind of getting ahead of myself here, right? And so alternative splicing is really what I was just talking about. You can produce different RNA, mRNA transcripts from the same gene. And depending on which exons you include in that final transcript is going to change what that protein looks like and its function, right? So here we have exons 9 through 13, right? And splice out all of those introns, so we get processed mRNA with exons 9 through 13. Now here we can actually exclude one of those exons by instead doing the splice from the end of exon 10 to the beginning of exon 12, right? That then gives us a different protein, which produces, a protein with a different function, right? And again, that's very much the same as what I just showed you with the example that I worked on. Okay. Oh, actually, I kind of jumped ahead a little bit. So before I forget, so really what I want to do is finish up this particular part of the PowerPoint here from 27 down, take a break, and then we'll come back and talk about what's up here, which is transcriptional silencing, right, which essentially comes after the start of transcription. So not too much to go through. So enhancers is essentially what we were talking about earlier, right? They're these regions which can be bound by activated proteins. They can be all over the place, right? There's no real rhyme or reason where these binding sites have to be. So also the presence of what enhancers you have can give you very different expression patterns based on what proteins are present in those particular types of cells. So this is a really neat example where essentially you have two different cell types, right? We've got a liver cell and a lens cell, right? You know, what's in your eye. And we have two different genes, right? So crystallin is a protein which during development clears your lens, right? So your lens is actually made up of a whole bunch of cells, and some of those are dead, right? And to make them transparent so light can get through, you have to clear all of the cytoplasm out, and that's what crystallin does. And albumin is I'm actually not entirely sure. I thought it was a structural protein. You get it in egg whites. But anyway, you don't need it in your eyes because that would be a really bad idea. So in the albumin promoter, right, the regulatory region, you have a different set of enhancers to the ones that you find in the crystallin gene. And so because those cells are different environments, essentially from a molecular point of view, you'll have different activators present in those two different cell types, right? So some may be shared, right? This gray one is an example, right? Is present in both cell types, but only the red and yellows are present in the liver cells, so you only get albumin expressed there, right? The crystallin gene does not have those red and yellow enhancer sites, so it is not expressed in the liver cell, and vice versa in the lens cell. You have a different set of enhancers, and so those are recognized by different proteins, and that gives you a different pattern of expression, right? This is the one way of thinking about the where, of gene expression. And then finally, insulators are essentially regions which will, when proteins are bound, inhibit the role of enhancers, right? So it could be by preventing a enhancer, an activated protein from binding. It could be preventing that bending of the DNA over so that that enhancer is not able to do its job. All kinds of different businesses. And then insulators. Oh no, I jumped. I'm sorry, I'm getting a little bit spaced. I kind of jumped along with it. Tell you what, let's take a break. I'm not like fully with it right now. And let's get back here. Hey, quit that. Let's meet back in 15 minutes. I'm going to go take some more painkillers. And then we'll get through the rest of the information after that, to be honest. Took a couple of aspirin. Hopefully that will help clear my head for a little bit. I think probably today's lab is going to be a little on the shorter side, just because I'm going to be in worse shape by then. Okay, so anyway, let's finish off where we started on. So I just reorganized this so it makes a little bit more sense. What we're talking about, well, let's talk about this here. So essentially, you can also have, so we talked here about enhancers, so that you have regions of DNA which proteins will bind to increase. Oh, let's get that back. Why is that happening? Ah, shoot. Sorry, computer's doing something funky. Resume share. Weird. That's where my chat box goes. Well, that's nice to know. Could do without this, to be honest, right now. Anyway, so talking about enhancers here, you have the opposite, obviously, which are silencers, right? And they may be either things which interfere with enhancers, right? So they reduce that activity. It may be that they kind of stop the function of the basal apparatus, right? So the RNA polymerase and transcription factors from assembling by physically occluding that. And they can also work in terms of, and this is a good example of that, of, let's get this a little bit bigger. There you go. I'm going to lose the chat, unfortunately. It's just gone and disappeared now. They can also interact with the chromatin modifications that we talked about earlier, right? So this is a good example here. We've got basically hormone or thyroid hormone is absent, right? And so we have this kind of assembly of various proteins and the like, and that results in the activation of an HDAC. Remember those histone deacetylases, right? And so that results in heterochromatin formation. Whereas when that thyroid hormone is present, that's that little T3 there, that results in the formation of the opposite, an activator complex, right, which involves histone acetyl transferase, which then results in the euchromatin formation, right, which you can see varying between these two here. right? So it's and this just gives you a window on how complex, right, that process can be, how many different things are involved and need to get together. And then the last little bit in this is this idea of insulators, right? So remember, again, enhancers can be bidirectional, right? It's not that they only affect genes in that direction. They can also flip that way as well. And so if you want to very much separate these two different genes, all right, and they can be going in the same direction or in opposite directions like so, you want to make sure that gene A is not turned on by enhancers for gene B, right? For whatever reason, they might need to be expressed in different places or in response to different stimuli. Then the way in which that is managed is through these insulators. And insulators are very much partitions between genes, right? So whatever is happening with gene B over here has nothing to do with gene A on the other side of that partition, right? So it's a way of kind of walling off chunks of the chromosome so that you can get a more fine-tuned response, right? You don't want to have inappropriate gene expression. Okay, so really all of that stuff, all of that from the cis-regulatory elements, alternative promoters, and you know, these different elements, enhancers, silencers, and insulators, plus alternative splicing, is a way of controlling what transcript is produced, right, at the end of the day, right? So whether that's the same transcript which is then processed differently, as in alternate splicing, or controlling where, when, and how much that transcript is produced at all, right? So that's really what we've been talking about so far, which is really, let's see where we're at, this bit here, right? Control of transcription and RNA processing. However, there's another couple of things. I'm not sure if we're going to talk about post-translation modifications. Yeah, that's there, but we'll mention that briefly. But in terms of making protein, it's not just how much RNA is produced, it's also how much RNA hangs around. So if you want to turn off gene expression, it's not enough just to silence expression here, right? Because you still have this transcript. You're still going to get translation from this mature transcript in the cytoplasm, right? So what you then need to do is get rid of that mRNA, right? You not only need to shut down expression from the transcription from the gene itself, but also get rid of any of the transcript from that gene that is still hanging around. Otherwise, you will still get protein being produced. If you got anything in the chat, give me a shout because I can't see it right now. It's Zoom is being a pain in the ass again. The only way I can do it is by it's not a mystery, like getting these things to work, and still they don't manage to do so. All right. All right, at least now I can see when anything pops up. Right, and so this is controlled by typically well, two things. One is the 3' UTR, right? So remember we talked about gene structure a while back, and we have the 5' and the 3' UTR, right? So transcript stability is influenced by the 3' UTR. And also a whole nother machinery. So there are non-coding RNAs that will bind to the 3' UTR, and basically they kick off the poly-A binding proteins. Oh, hang on. Yeah, this is still PowerPoint 18. It's just moving around a lot. I forgot to reorganize these PowerPoints before I posted them up. So I'm sorry about that. They're kind of all jumbled around. So yeah, we're on slide 5 of PowerPoint 18. Anyway, so there's that aspect, right? We're not going to talk about that now. What we're going to look at is what's called RNA silencing, right? And this is actually a super, super cool technique that I use a lot in, or used to, at least when I was able to, use a lot in my lab. And so there are two different kind of purposes for this, right? And that determined which one is being used. One is to protect against RNA viruses, right? So we have, eukaryotes have a mechanism to, like an intrinsic mechanism, right? This isn't part of your immune response. This is actually part of your cellular response to double-stranded viruses. And what they do is they recognize those double-stranded RNA sequences as being exogenous, right? Wrong, basically. And then they use an enzyme called dicer, right, which dices up that double-stranded RNA and causes the assembly of an RNA-induced silencing complex, or RISC, right? And its job is to use one strand of that mRNA to go around and basically look for any other bit of mRNA that matches. And as soon as it finds it, it cuts it, right? So if your cells are infected with a double-stranded RNA virus and it promotes this mechanism, that's a way of getting rid of that RNA virus, like its actual genetic material, before it kind of kicks in. Now we also have another pretty neat system, and these are called small interfering RNAs. See if I can get this typed up. Oh, this is just so fucking annoying. I can't get to the chat while I'm sharing. God damn it. Fucking stupid. So these small interfering RNAs are actually you can use them as a very powerful technique in something called RNA interference, right? So this actually won Andy Fire and Craig Daniels, I think. I can't remember his name. Got a big chin. That's all I can remember of him. Won them an Nobel Prize for their work in C. elegans for RNA interference. And so you can literally just feed worms bacteria expressing this double-stranded RNA, and it will shut down gene expression of that gene, right? So it's a very, very powerful, very useful technique to use. And this is actually where science kind of merges into magic, as far as I'm concerned. So the worms eat bacteria because that's what they eat anyway. It's basically what they can stuff in their mouths. And then the double-stranded RNA that was in those bacteria gets out of them, right, as they get ground up and digested, and it gets through the intestinal wall of the worm, and it spreads throughout the worm. And then, apart from the neurons, which are kind of impervious to RNAi, and then basically shuts down gene expression. And it does such a thorough job that for some genes, like those involved in early development or the cell cycle, it results in no worms, right? So you can put adult worms on this RNAi and you'll get no offspring. All of those eggs will be sterile, they'll be dead, right? So it's an astonishingly powerful technique. And it's really essentially, like a lot of these techniques that we use in biotechnology, it's really taking something which is kind of an intrinsic process, right, which is normally just used by cells or bacteria or whatever to do their daily lives, and then putting it in a different context and using that to ask scientific questions, right? So a lot of what I do is knocking down gene expression of different proteins or different genes, really, and then seeing what effect that has on a particular phenotype that I'm interested in. Might be cell death or lipid metabolism or developmental biology, right? So those are all different questions that I've been able to ask using this. Now, the other version, right, is, and this is not mRNA, it's miRNA, and that's microRNA. And so essentially what this is small endogenous, right? So those are produced by the cell themselves, non-coding, spelling not working well, RNAs that inhibit translation. So let me get back to this. And so it's a similar kind of process. It's not quite the same, but functionally the outcome is similar. And so what happens here is that instead of having this double-stranded RNA, now we have a stem loop structure, right? So we talked about these in control of transcription termination prokaryotes. You can see them in tRNAs, right? So lots of RNAs form these different structures. And so essentially what happens is that that gene that encodes this non-coding RNA or this microRNA is expressed. And as soon as that transcript kind of comes out, it folds into a stem loop structure. I can't do a loop, like the loop on top gets cut off and then the same principle applies, right? This RISC complex, this RISC complex basically does the same job. It scans, right? It uses that little single-stranded bit of RNA as a barcode. And it basically just kind of goes around the cytoplasm until it finds an mRNA that matches. And when it does, it binds to it and it inhibits translation, right? It inhibits the typically how poly-A binding proteins promote ribosome assembly, right? And so yeah, the outcome is the same, right? It doesn't necessarily degrade the mRNA the way that siRNAs do, but it results in the same outcome, which is no translation of that mRNA. And then just to finish up, let's check if anyone has any questions. Just to finish up, go over here. There's also a whole bunch of other stuff, right, afterwards, right? I'm not going to talk about it very much because we just don't have very much time. And that involves processing proteins such as insulin, right? It needs to be cut up by other proteins to be functional. It could mean having other things added to it, right? So a lot of the proteins that end up on your cell membrane have sugar residues added to them. So they become glycoproteins, right? For example. And even proteins which are kind of like the right shape and functional also can be modified to turn them on or turn them off, right? So proteins themselves have kind of built-in switches which allow those proteins to be controlled, right? So you could have a lot of protein of a particular protein, but if it isn't activated, it's not going to function. And so you can also control essentially the function of a protein independent of how much of that protein is being produced. Right, and so quite often that controls enzymatic activity. Kinases are a good example that add phosphate groups. They're typically activated by other kinases. You can also have transcription factors, right? We talked about these a bunch earlier on. And many transcription factors are not present in the nucleus, right? And so they're only imported into the nucleus when they're activated. Right, so again, you have this extra layer of control on top of all of that other stuff. It is truly mind-boggling, right? The degree of complexity that exists, right? And I'm not sure any one person can really truly grasp all of it, to be honest. It's like I don't know, the knowledge of everything in a sense. It's kind of hard to get over that. And also, once a protein has been produced for various different reasons, it can be destroyed. So cell signaling, for example, when you have a signal that says, hey cell, you need to do this, whatever this might be. Quite typically, once that signal has been received, the receptor, the protein that receives that signal, is destroyed. Right, and so that essentially self-limits that kind of signaling process, right? So there's an awful lot of stuff going on. Maybe proteins are damaged, they're not needed anymore. Kind of the way that red blood cells are recycled. You know, they only live for like a week or so, 4 days, 5 days, something like that. Proteins are also recycled, right? If they're no longer needed, or maybe they've started to accumulate damage. Okay, so that's pretty much it for control of gene expression in eukaryotes. There's a lot of stuff here, a lot. So probably a little bit difficult to kind of process it all right now, and that's fine. But do please go through this over the weekend and bring any questions you have to class on Tuesday, because that's really going to be our last chance to talk about this before the exam that's going to be on next Tuesday, Tuesday the 20. Is that the right date? Yeah, Tuesday 28th. I thought that was the right date. Teaching on Tuesdays and Thursdays is weird because I think it's Friday and it's not. Right, that's pretty much it from me for this PowerPoint. Just want to take a little break if anyone had any questions before we go on to the next one. But do try and keep in mind that kind of linear series of steps that occurs between DNA, right, and its chromatin structure, and functional protein. And at every one of those steps, there is a way of moderating that step or modifying it to control the total amount of protein produced, right? So how much protein, functional protein, and that's important, not just how much protein, but how much functional protein you get is the consequence of all of those, right? So it doesn't matter how much, transcription you have, how stable the mRNA transcript is, if the protein produced needs to be phosphorylated to be active and it isn't. Effectively, you do not have any functional protein. You may have a whole ton of protein there, but it's not doing anything, right? And so that's really what we care about. So try and keep that in mind when we think about all of those different steps, right? How much functional protein do you get out at the end? All right, so I've got a PowerPoint here, and that's essentially what this is a, pardon me, kind of a way of using a lot of these discoveries, right? These processes to ask questions of our own. So not necessarily anything to do with that process, but really just using that process to ask questions about something else completely unrelated, right? And I would want you to read through this, right? It's basically talking about DNA modification, PCR, which we talked about a bunch already with the lab, sequencing, and then talking about cloning, right? So some of this material is a little on the old side, right? We're talking about restriction enzymes, which come from different bacteria, and they recognize specific sequences, DNA sequences. But we can use that to be able to basically cut out or generate bits of DNA which will stick together, right? So, for example, you know, if you generate 2 bits of DNA that both have a Hind 3 or Hindi 3 restriction site, then when you cut them with that enzyme, they will stick together really nicely. And you can see that down here. Also, this is also kind of fun because this ties in a little bit of what we learned way back when we talked about DNA replication. Any of you recognize this enzyme? You're gonna have to shout out if you do, because I can't see the chat session for some reason. This is the enzyme that seals the nicks in the backbone at the very, very end of DNA replication, right? So in this context, what we would use that to do is to stick together different bits of DNA to create one final functional piece of DNA, whatever that might be. So I'm not going to go through, if you want me to go through any of this, that's totally cool. What I want to do is actually just go back to that paper that I showed you earlier, because it really encapsulates all of this, right? And this is really what, you know, I've done for a long time, right? I don't do it so much anymore because I don't actually need to for a lot of the questions that I want to ask, but it just depends on what questions you're asking, really. Now, where has that gone? Here we go. So one of the really cool discoveries, and I'd love using this particular discovery, is that of green fluorescent protein, or GFP. That's what and that's why it's in colored green, which is super cool. And so that was from a jellyfish called Aquia victoria. I don't remember where that was from exactly, maybe the Caribbean. But essentially it glows, right? So if you've ever seen pictures of, you know, those really, really beautiful jellyfish that kind of fluoresce, they have all kinds of different colors as they kind of do their jellyfish thing, right? Those are caused by proteins, fluorescent proteins, right? And so GFP is a small, fairly small protein. And when you shine blue light on it, which in jellyfish is generated by another reaction involving calcium, we use just a blue light on the microscope. When you shine blue light on that protein, it absorbs that blue light like chlorophyll does, essentially, and it re-emits it as green, right? So typically fluorophores always re-emit at a higher wavelength, right? So blue is like 450, something like that, give or take. Green is 580, I think, nanometers in terms of its wavelength. So anyway, now we have this awesome tool, right? So now we can use that protein by sticking the DNA that encodes it onto other bits of DNA, right? So one of the big jobs that I had in this particular project is trying to figure out where these different promoters were active, right? So it's all well and good knowing that we have these 3 different promoters, and this one was actually discovered as a result of doing this work. But where are these expressed, right? What's the functional consequence of having these 3 different promoters? And if you've got any questions, just, tell me to stop and I'll explain stuff. And so essentially what we do is, well, what I did is the same as what you would have done with the flies. I got a whole bunch of worms. I told them that their sacrifice would be very noble. Actually, I didn't do that. I just put them in a tube and ground them up. But I got their DNA out and I amplified the whole chunk of it, right? Which is why it's called genomic, right? So as much of the promoter as I could get there, and all of the coding region, right? So all of the different promoters, all of the different exons. Yeah. So in terms of the gene, it was in, as best as I could make it, its genomic context, right? I wasn't kind of taking little bits out and sticking them in some weird context. It's all exactly as you would find it in worms, right? And then I cut that and I stuck, using those restriction enzymes and ligase, the coding sequence for GFP, and I just stuck them together so that they would form one large protein, right? So this exon 4, when translated, would translate straight into the GFP coding sequence, and I would make a new protein, transgenic protein, which would be both PNC1, which is the protein I'm interested in, with an extra bit of protein stuck onto it, which would fluoresce when I shine blue light on it. Does that make sense? Hopefully. Now there's an awful lot of fancier way of doing this where rather than taking that chunk of DNA out and then sticking it with a GFP, now you would use CRISPR-Cas9 to put the GFP in to the actual genome of that organism exactly where you want it in terms of a translational fusion, right? So same consequence, bit more accurate, right? Because you're not, you're only dealing with the 2 copies then. You're not dealing with extras. Okay, so once you make that, and it's a called a plasmid, right? So essentially what you would do is you would stick that gene of interest, whatever that might be, in here, right? So that you can grow up this bit of circular DNA called a plasmid. Yeah, there we go. It kind of looks like so. In bacteria, and then what you do, and this is, it sounds really interesting, and it's not. It's bloody hard work and boring as batshit. You then inject that DNA into worms. And so these are worms that are, where's my camera? About a millimeter long, give or take. And so you use a very, 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 very fine needle and a very steady hand. And you inject some of that DNA, and in some of their offspring, that DNA is present in those cells, right? Now let's just go and see what that looks like. Actually, I wonder if I've got, now that'd be too much of a pain in the ass trying to find the original pictures. They're on my hard drive somewhere. Let's make that a little bit smaller. Where's the genomic? I think that's D. So now you can use a what's called a laser scanning confocal microscope. So this uses a laser to excite that fluorescent protein. And it also takes pictures in slices, right? So a worm is a 3D object, right? And you have cells kind of within that object. If you imagine the light's coming from below and then your eyes are at the top, right? So it's very hard to see kind of all of those objects distinctly using a normal microscope like a epifluorescence microscope. So instead what you do is you use a very expensive microscope. They're about $500,000, give or take. And you use a laser to excite just this level. And then you do this one, and 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 this one. And depending on how thick those slices are, these are assemblies of typically about 30 images, right? So they form what's called a Z stack or Z stack. But what it allows you to do, this is the white light image over here. So this is a worm's pharynx. You can see those are essentially its teeth, believe it or not. This is a grinder. So it's like a turkey basting bulb, to like suck up juices to spray over the turkey. This sucks the bacteria in here, and then it has these kind of teeth which just grind up the bacteria before they swallow and go into their intestine. And so around this area, you have a worm brain, basically, called the nerve ring. And so what you can see here is, let's see if I can blow those up a little. Ooh, it gets all kinds of pixelated now. Let's not do that. Can you all see what I'm pointing at? This long process over here, that's actually a neuron, right? So this is probably a dendrite. Yeah, this would be a dendrite. And this actually goes out to a sensory pore, essentially a worm's nose, right? And when you look at these cells, I can't remember what they are. It's in the table kind of further up in the paper. The very bright dot is the nucleus of that cell. Sorry. And then the paler area around it is the cytoplasm, right? So you can basically, and these are tail neurons, right? So those are kind of around here, right? And you can see another projection kind of coming out. It's slightly out of plane, unfortunately, for whatever reason. Maybe the Z stack wasn't deep enough. But here's another probably dendrite process, which actually it's involved. I'm not actually sure what tail neurons are involved in, to be honest. Right. So you can actually just see, right? Literally physically see where this gene is expressed, right? Essentially, it's in all of these cells, right? And so when you do this, and this was about 4 years work, give or take, this whole paper. Not solely, there was other stuff going on as well. And for a little while, at least, I don't remember most of this stuff, you can actually use that to identify cells, right? So for example, the genomic version is expressed in all of these neurons in the head, right? And they used to do different things. Some sense temperature, some sense chemoattractants, various different things. And you can just go through and you can catalog, right? And that ties into a function, right? Why is this gene expressed in these cells? Presumably it's doing something in these cells which is necessary for those cells to do their function, right? It's also expressed in the intestine, which is kind of interesting. Now, let's just see if I can get. Awesome. Oh, I do have the chat back. Fabulous. Now, you remember that I talked about how we have 3 different promoters, right? And so now what we can do is, I'm not going to talk about that one up there. Now we can look at all of those promoters individually, right? So before we looked at the whole context, right? Where is all of the sites of expression, right? But that doesn't tell us what those individual promoters are doing, where they are expressed. And so now what we can do is we can essentially take each promoter. So we've got, if we go up here, for example, right, we can take this promoter and we can take the essentially the, trying to think of a good way of describing it. We can take all of the exons, stick them together so we don't have any of these introns, and then we can fuse that with GFP, right? So we can do 1A promoter with 1A, 2, 3, 4. We can do 1B promoter with 1B, 2, 3, and 4. And we can do 1B2 promoter with 2, 3, and 4. And stick those all with GFP and then look at the differences. And that's essentially what these three different constructs are. And so when you look at that, oops, let's get back again. Why did I can't remember why I did it this particular way around. Who knows? And so, for example, this is the 1A promoter, right? There's expressed in a whole bunch of muscle tissues and can't remember what these cells are at around the side. But when you look at the next promoter along, exact same part of the worm, right? So we've got head of the worm there. We got head of the worm there. Now we get a very, very different pattern of expression, right? Instead of being just, you know, these pharyngeal muscle cells and, you know, other bits and pieces, now it's expressed in all of these different neurons, right? So when we looked at that whole big picture and we saw all these different sites of expression, some of those are purely from one promoter. Others are purely from another promoter. And that's what we're able to then kind of really drill down into and understand. And then this one over here looks very different again, right? And so now you can start getting a much bigger picture of what is going on, right? What are all the different things that are happening with regards to this one single gene, right? This is just one gene, you know, out of 20,000, right? And now you can see just at the level of transcription how astonishingly complex this can be. And now you see, so here's the head over here. This is the intestine, right? So now we have a completely different site of expression, which you don't see either in this worm, right, which has the first promoter, or this worm, which has another promoter, right? So, and you can even, when you look at it, where are we at? Oh, it's over here, right? You can even see the secreted protein, right? So these are gaps in the worm, right? They're filled with fluid, extracellular space, basically. And these are actually all eggs. So these are, they're not eggs, well, they are eggs. These are actually embryos that are developing. You can see all the different nuclei that will eventually become a worm. Well, this protein is now present in the spaces between those eggs. It's not actually in cells, it's in the extracellular space. So now you can actually start putting together a much bigger picture, right? Just from looking at stuff, right? This is really, I mean, there's a lot of other work that particularly other people did in terms of function and phenotypes and, you know, processes that it's involved in. But just from looking at this stuff and spending hours and hours and hours with my eyeballs glued to a microscope, you can actually get a much more complete picture of what this gene does. right? And it's a lot more nuanced than one would expect. And actually, this finding here, the fact that it is secreted and functions in cells that it's not present in, was a really big finding for this particular field, because all the other versions that have been discovered so far only worked within the cells that they were expressed in, right? This one is able to provide function to cells where it is not expressed. So if you're really into like that whole NAD metabolism thing, that was a really exciting finding. So really, this is just a snapshot. I don't want you to kind of like get totally into this, but this is just a really neat way of showing what you can do with some of these techniques, right? With CRISPR-Cas9, right? So let's go back up. Those are just graphs, they're kind of boring, right? CRISPR-Cas9, rather than using a mutant screen, which is where this allele came from, now you can start actually creating your own mutations, right? Specifically, like very particular ones, right? So this is a signal sequence mutant, right? I created it, but I couldn't look at it in the worm itself, right? In its original context. This was another construct which I looked at. Now you can do that. Right? It wasn't really a technique that was widely available when I did this work. Now you can actually knock out that signal sequence and see what consequence that has on worm physiology and reproduction and the like. You can also get an RNAi clone to this, right? And you can feed worms this, and you can actually reduce the amount of functional PNC1 protein, and have a look at the effect of that on physiology and metabolism and the like, right? So there are an awful lot of really, really, really cool techniques that you can use, essentially using stuff that we know about genetics and molecular biology, right? It's not that we're really reinventing the wheel, right? We use natural selection and evolution basically to do all the work for us, right? It's that old adage of, if you want to make something small fly very well, copy a bird, right? Or an insect or something like that, right? You don't have to reinvent the wheel. And really a lot of molecular biology is about taking what we've discovered just from stuff being interesting, right? Wanting to know how it works, and then using that to ask questions of our own. And that's really one of the, in my opinion, is one of the most exciting parts of molecular biology. It's kind of the use of it to do stuff, right? And it can have really kind of life-changing consequences too. Insulin is always my favorite example, but for example, the COVID-19 vaccine that the University of Oxford is working on is an mRNA. It's an RNA vaccine, right? So I'm not actually quite sure how it works. I don't know if it results in the production of proteins or if it induces that RNA interference pathway, because COVID-19 or coronavirus is an RNA vaccine, is an RNA virus, sorry. I'd have to have a look into that to see what the mechanism is. So there you go. It's some pretty cool stuff, and you can ask some really neat questions as well as a result. So I'm going to take a break now. We can meet back at 2. Not going to spend a lot of time on lab today for a variety of different reasons, one of which I'm starting to flake out quite badly. But also because Dr. Page has done such a good job of making those videos for all of you to use, that really anything I say will just be a duplication of that. So next Tuesday, we will be looking at genomics, right, which is really like the large scale picture of genetics, right, which certainly the work that I've just talked about now is very much the small scale of genetics, right? Looking at an individual gene and how it works. Genomics is looking at whole genomes and genomes between species and individuals. So we'll be looking at that on Tuesday, and also we'll have time, if any of you have any questions about the material, for me to be able to go through those with you before you take your final exam. Now, one question that's probably on your mind is it a cumulative exam? And the answer is no, it is not. So exam 4, also known as the final, is on only material we've studied since exam 3. Although again, as always, as you can see here, stuff pops up that you have already studied earlier on in the course, like DNA ligase, for example. So inherently, it's a little bit cumulative, but it's not cumulative in terms of the material that we've studied that you will be tested on. And don't forget, that is on Tuesday. Does anybody have any questions about that or the exam or any of this material that we've just covered? All right. Yeah, I hope I feel better too. I'm probably just going to maybe go out to the pharmacy and get some of that. What do you call it, Angela? Was it dark rum? I think that might help with my headache. Bayer, okay. I can go and find some of that and neck some of those. But anyway, let's meet back at 2, Bayer back and body pain. I remember that. I just got to go in and say, oh, my back and body pain, and they'll go, that's the one. So let's meet back at 2, have a little bit of a longer lunch than usual. And I don't envisage it taking very long. Really, I'm just going to be pointing you to the different things that you need to be doing and your resources for doing so. Next Tuesday, we can certainly go over any problems that you have doing any of that stuff. So let's meet back at 2. All right, ladies and gents, thank you for your time. I'll see you in a little bit.